Hi, and welcome to a new series, Med with Maram Shorts. So, we'll be starting with types of capillaries, and there are three types of capillaries. So, the first one is continuous, and basically, the idea of a continuous is that there is a continuous basement membrane as well as a continuous endothelium. So, the basement membrane as well as the cells, the endothelium, are all continuous. So, they're covering the entire capillary, okay? And this type of uh, capillary only allows substances to go in and out by diffusion. So they're the most common type of capillary and they're found all over the body. Their idea is that they're only allowing substances to go in and out if the substances are small enough to go through diffusion. Second type of capillaries are fenestrated capillaries. Now, fenestrated capillaries have a continuous basement membrane. However, they don't have a continuous endothelium. Their endothelium has perforations. So these tiny little perforations or fenestrations are there to allow exchange in and out more. So it's in areas where you find lots of exchange. So for example, in the intestines, in the kidneys and endocrine organs. So for example, let's take intestines as an example. You'll be absorbing a lot of carbohydrates, a lot of amino acids, and so on. And to be able to absorb those, you'll need the fenestrations for better absorption. In kidneys, you're trying to filter as much as possible. And in endocrine organs, you're trying to get the hormones out. So you'll be needing fenestrations. The third type of capillary is sinusoidal capillaries. And sinusoidal capillaries are really different. They have a partial basement membrane, so the basement membrane is not continuous. They have really large gaps in the endothelium as well, so they're really big open areas. And the idea here is that it's for areas of exchange of macromolecules or blood cells. So we're exchanging a really big substance, like for example, plasma proteins. So in the liver, for example, when you're forming a plasma protein and you're trying to put it in the blood, you're going to need a big gap because fenestrations are not big enough. So you're going to need a really big gap to put in the plasma protein. For the bone marrow as well, when you're synthesizing a red blood cell, for example, you'll need really big gaps to be able to put it in the blood. And for the spleen, it's the other way around. You're trying to get the red blood cells out, so you need really big sinusoidal capillaries. So always think of them in areas where the blood is involved. Either you're forming plasma proteins or you're dealing with the blood, such as the bone marrow and the spleen.